Students, welcome you all on the behalf of Rahul Anand tutorials. So as you know that I am Rahul Anand and this is somewhat around my channel whose name is actually the Rahul Anand tutorials. So now let's begin with the very interesting topics which are often being asked in terms of your NEET, often the AIIMS, often the All India Pre-Medical Examination. So these are literally belonging from which of the chapters of your book? So these are belonging from the here, minute of the cell, the unit of life. So today we are going to study about what? So here we are going to study about the fluid Mosaic model of the plasma membrane. So your most accepted proposed result with reference to what? With reference to this minute of the fluid Mosaic model was proposed by whom? So it was proposed by the two scientists who was actually what? So these were actually the here minute of the Singer as well as the Nicholson. So when we are talking about the era in which they actually proposed the result of this in the fluid Mosaic model. So it was around how much? So it was around 1972. Now, often when we are talking about these cases, means what? Here, mean the fluid Mosaic model. So your fluid Mosaic model are often known as what? So your fluid Mosaic model are often known as the here minute of the protein Mosaic model. Why is it so? Remember, so we are having the broad kind of the codons when we are talking about here in this case of what? Of the here minute of the fluid Mosaic model. So we are having what kind of the proteins here? So here we are going to have the integral protein as well as the fibrous protein. Remember, often we are having the intrinsic protein as well as the globular protein. When we are talking about the integral protein, so it will be present where? Find here. So it is the portion where somewhat the integral protein will be present. Often when we are talking about this case, so its composition with reference to your overall protein present in terms of what? Present in terms of the fluid Mosaic model is around how much? So it is around 70% of the protein. When we are comparing the case of the 100% of the protein. Thus, these are often present in terms of what? In terms of the wider ratio. So you could often find here on the board. So these are particularly present in terms of what? In terms of the wider ratio as well as in the just like the middle portion occupying as well as these are particularly penetrated with reference to what? With reference to the phospholipid bilayer with reference to how much of the ERA as well as the concentration around 70% of the concentration. Thus, these are particularly being penetrated. Thus, particularly, it could not be separated with reference to what? With reference to this model. Fine. Often, when we are talking about these cases, means what? When it comes the fluid Mosaic model. So you have to understand the certain facts in it, such as when we are talking about the case of the fluid Mosaic model. So in this particularly, what is happening? So in this, we are going to study about the ultra structure as well as the complex structure with reference to what? With reference to the plasma membrane. So remember about the case of the plasma membrane. So these are what? So these are the thick elastic. Often what? So these are the thin elastic membrane, often known as the selectively permeable membrane. Why is it so? This is due to because in this case what will be happening? So in this case the particular concentration gradient with reference to the entering of what? Entering the substances in terms of your cell is particularly taking place. Often what? Often the existing mechanism or either the existing of the concentration mechanism from the cell toward what? Toward the external environment are literally being taking place. And here, the mechanism is particularly being employed with reference power. So it is being employed with reference to the two of the phenomena, such as what? Such as the osmosis as well as the diffusion case. Often when we are talking about the here minute of the transportation of what? In order from exchange of the material, either from what? Either from the cell towards the external environment, or either the vice versa is occurring. So it does not require any of the amount of the energy. Fine. So, if it does not require any of the amount of the energy, how is it that the overall process are literally being employed? In terms of what? In terms of the transportation mechanism. With reference to what? With reference to the selectively permeable membrane. So, these are taking place with reference by the help of what? By the osmosis as well as the diffusion. Remember? So, when we are talking about the osmosis case, so in this case, what will be happening? As well as when we are talking about the case of the osmosis, so in this case, what will be happening? So in this case, the exchange of the water molecules will literally take place with reference to what? With reference to the concentration gradient, either from what? Either from the cell towards the outer environment, or what will be happening, or either the vice versa. Remember, so your cell are particularly often having the excretory mechanism, as well as it often requires the particular amount of the water in order to tune what? In order to tune the cell organelles. Remember about the case of the cell organelles. So you are having the different types of the components with reference to your cells. So these are actually what? So these are the nucleus, the centrioles, often what? The plasma membrane, endoplasmic reticulum, then the Golgi complex as well as the Golgi apparatus, as well as what? As well as in this case, we are often having the certain more components such as the lipid, often what? Often the centrioles as well as the vacuoles, etc. After that, what will be happening? So after that, we are having the second case in terms of what? In terms of the diffusion. Remember about the case of the diffusion? So the exchange of the gases will literally occur as well as take place with reference to what? Either from the cell towards the outer environment, or either what will be happening, or either the exchange of the gases literally from outer environment towards what? Towards the cell region will be taking place. So these are coming under what case? So these are coming under the osmosis as well as the diffusion case. Now, and when we are talking about the case of the fluid Mosaic model, so you have to understand the certain more facts in it, such as what? So in this case, the intrinsic protein will particularly play an important role as like what? So it will be particularly floating as like what? As like the iceberg floating in the sea of the lipids. So you are very clear as well as understanding about that means what are actually the lipids. Remember, so lipids are what? So lipids are actually the natural occurring substances. So these are the natural occurring substances which are particularly restricting as well as insoluble in water. So these are insoluble in terms of the water molecules. Often playing important role for what? Often playing important role to maintain the healthy status with reference to your cell, as well as what? As well as to maintain the here in order to provide the rigidity with reference to what? With reference to your cell. Just like what? Just like the case of the plasma membrane. So these are often known as what? So your plasma membrane are often known as the here meaning of the cell membrane. As well as in this case, you will be often having what? So in this case, we are often having the globular protein. The role of the globular protein is just like as what? So here the globular protein will play an important role as like the role of the enzymes. Remember about the case of the enzymes. So what are enzymes? So enzymes are the biocatalyst, playing important role for what? Playing important role to increase as well as decrease what? Playing important role to increase or decrease the rate of reaction without being altered in overall reaction. Remember? So these are quite bit the case of the enzymes. Often these are known as what? So your enzymes are often known as the biocatalyst as well as what? So these are often being further divided into the two broad categories, such as what? Such as the apoenzymes as well as the cofactors. Remember? So one will be the proteinous enzymes. On the other hand, we will be having the non-proteinous enzymes. Often when we are talking about in this case of the fluid Mosaic model, so we are often having the extrinsic protein. Remember about the extrinsic protein as well as the intrinsic protein. So overall mechanism with reference to the whole protein, consequent with reference to how much of the percentage in terms of the 100%. We will be having the extrinsic protein as well as the intrinsic protein. So your intrinsic protein are often known as the integral protein. The consequent with reference to the protein is how much? So it is around 70% of the overall out of what? Out of actually the 100% of the protein. Now, so these are particularly deeply penetrated, of course. Now, so let's understand about this in terms of the detail. So you are very clear about that means what are actually the intrinsic protein as well as the integral protein. Remember? So in the consequent of what? In the consequent out of the 100%, we will be having the composition of what? Actually the intrinsic protein as well as the integral protein. And actually in terms of what? In terms of the 70% of the ratio. Often these are particularly penetrated with reference to the phospholipid bilayer. Thus, these will particularly cannot as well as are not able to escape from what? Are not particularly able to escape from the phospholipid bilayer. Thus, these are particularly penetrated with reference to the wider amount, often consequenting about how much? About 70% of the total protein. Now, so these are often known as what? So these are often known as the icebergs floating in the sea of the lipid. Means what? Means in terms of the smaller consequent. And these are actually what? Mean in terms of the smaller consequent. So here, smaller consequent will be around 30 as well as in this case what will be happening? So in this case some of the integral protein will particularly penetrate what? Will particularly penetrate the phospholipid bilayer. And after that what will be happening? So these are particularly being supported with reference to what? With reference by the help of the tunnel protein, then the transmembrane protein. So you are very clear about that means what are actually the tunnel protein and transmembrane protein. Remember in the case of the first classes of the fluid Mosaic model we have understood about that. So these are the composition structure. Often these are what? So these are particularly the pores-like structure find here on the board here. As well as the transmembrane protein are playing important role for what? So these are playing important role for the passage of the substance. Often when we are talking about these cases in terms of the tunnel protein as well as the transmembrane protein, in terms of the pores, so these pores are what? So these pores are not permanent. Often when we are talking about the case of the tunnel protein as well as the transmembrane protein, so these will be particularly existing in terms of the pair. So you could often find here on the board. So these are particularly in what direction? In terms of the pair direction. Often in these we will be having the what? The tunnel protein as well as the transmembrane protein in terms of what? In terms of our fluid Mosaic model. So you could often find here on the board. Often we are having the different types of the components such as the integral protein. Then what? Then the transmembrane protein. Often in turn we will be having the extrinsic protein. Then what? Then the lipids, the sugars, the glycoprotein. Often the extracellular matrix as well as the extracellular species. Often what? Often the phospholipid bilayer, etc. Now, so after that we are often having the composition as well as the important role with reference to what? With reference to the carbohydrate. So here, what is the role of the carbohydrate? Now let's understand about it in terms of the brief. So here, carbohydrate are literally present in terms of what of the ratio as well as the percentage. As well as when we are talking about the case as well as the role with reference to what? With reference to the case of the carbohydrate. So here we will be often having the carbohydrate in terms of what? In terms of the fluid Mosaic model. So in terms of how much of the ratio as well as the percentage. So around how much of the percentage? So around 1 to 8 of the percentage of the carbohydrate are literally being present in terms of what? In terms of the branched as well as the unbranched chain of what? Into the branched as well as the unbranched chain of the glycoprotein. So remember, so glycoprotein is often being present in terms of what? In terms of these, of the here level diagram of the phospholipid bilayer. So we are often having the carbohydrate, and here the role of the carbohydrate is what? So it will be present in terms of what of the ratio or rather the percentage, around 1 to 8 of the percentage of what? Of the branched as well as the unbranched form of the carbohydrate are literally being present with reference to the glycoprotein. But when we are talking about the case in terms of what? In terms of the role of this carbohydrate in terms of here, in terms of the fluid Mosaic model. So you are having the different types of the composition such as the integral protein, then the intrinsic protein. Often you are having the glycoprotein as well as what? As well as the carbohydrate. So these carbohydrates are playing important role in terms of the recombination as well as the recognition mechanism. Remember? So in this case, what will be happening? So your cell will particularly recombine in order to form the tissue. Remember? After that, your here, in terms of the recombination of the bacteria as well as the antigen will be occurring. When we are talking about the case of what are actually the bacteria, remember? So what are bacteria? So these are often being further divided in terms of the two broad categories such as the gram-positive bacteria as well as the gram-negative bacteria. Often these, in terms of the gram-positive bacteria, are having what? So these are often being divided in terms of the aerobic bacteria as well as the anaerobic bacteria. So fine, the one which will be growing in terms of the presence of the oxygen coming under what case? So it will be coming under your aerobic bacteria. Often when we are talking about the here in terms of the anaerobic bacteria, the one which will be growing in terms of what? In terms of the presence of the carbon dioxide or either in the absence of the oxygen coming under what case? So it will be coming under your anaerobic bacteria. Often when we are talking about the case here, means what are actually the antigens. So fine. So here recombination as well as what? As well as the recombination of the antigen will be occurring. Fine. In terms of what? So these are quite a bit the role of the carbohydrate. After that, we'll be understanding about what are actually the carbohydrates. So now, when we are talking about the case of the antigen, remember? So antigen are what? So antigen are the foreign agent. Antigen playing important role for what? So antigen playing important role to excite the what? To excite the antibody, to form what? To form the antibody molecules. Remember, so antigen are what? So these are the foreign agent as well as we are having the antibodies. So these are what? So these are particularly being performed as well as specialized with reference to the certain of the cases. So as like the antigen play important role to produce what? To produce the pathogen coming under from the external environment with reference to code as well as what? As well as destroy our body. After that, our antibody will particularly play important role to provide the certain types of the here in terms of the enzymes. So these often play important role to secrete the vital types of enzyme. As well as we are often having the different types of the vital types of the cell play important role to coordinate with the antigen. So antigen are what? The poison molecule. So it could be what? So it could be helpful as well as harmful in nature. But generally in these cases we will be having the harmful often. So we are having the different types of the antibody responses. These are what? So these are the helper T cell, the killer T cell, the suppressor T cell as well as the memory T cell. Remember about the case of this? So memory T cell particularly revives the all the events such as what? In terms of the event of the video. So process it as well as preserve it. So suppose that we are having the vital types of the coding such as the symptoms of the cholera. So our cell will particularly remember what? The particular type of the medicine as well as the whole sequence, that how it was particularly being prescribed, just like our brain. As well as we are often having the suppressor T cell, particularly playing important role to suppress with reference to what? With reference to the here main term of the antigen. Then what? Then we are often having the memory T cell, the killer T cell. So killer T cell will be playing important role to kill by secreting the certain types of the substances as well as the here in terms of secreting some of the vital types of the organic components. As like we are having the codex in terms of the lysosomes. Remember the case of the lysosome. So these are the suicidal bags. Now, so this will be playing important role. Fine. After that, we are often having the carbohydrates. So when we are talking about the carbohydrates, so what are actually these? Tell me. So carbohydrates are the optically active. What? So carbohydrates are the optically active polyhydroxyaldehyde as well as the polyhydroxy ketone. Often when we are talking about the composition as well as the different types of the functional group which are particular, as well as when we are talking about the case in terms of what? In terms of the carbohydrates. So carbohydrates are what? So carbohydrates are the optically active polyhydroxy ketone as well as what? As well as the polyhydroxy aldehyde. Now, so when we are talking about these, means what are actually the what? composition as well as the chemicals literally present in terms of the formation of these one, the carbohydrates. So these are what? So these are the carbon, hydrogen, then the oxygen. So find here. Often when we are talking about what are actually the here functional group which are generally being involved in terms of the formation of the carbohydrates. So these are what? So these are the aldehyde, ketone, then what? And the hydroxyl group. So find here. So this is the somewhat condition when we are talking about the case of these, means what are actually the role of the here in terms of the carbohydrate. Often we have read about the bit introduction, means what are actually the role of the carbohydrate in terms of what? In terms of actually the fluid Mosaic model. Often we are having the different types of the components as well as the protein in it. So fine, as well as remember, so we are having the intrinsic protein as well as the integral protein. So these will particularly resembling as well as present in terms of the same codings with reference to the membrane space. In terms of what? In terms of the phospholipid bilayer as well as in terms of the particular Mosaic model. But when we are talking about the case of the globular protein, so your globular protein will particularly present in terms of what? In terms of the separate portion. Fine. As well as when we are talking about what are actually the globular protein, remember? So these are what? So the protein, tell me. So the protein which are quite bit soluble in terms of the water molecules coming under what case? So it will be coming under your globular protein. Example are what? So the example are the pepsin as well as the renin. Fine. Playing important role to digest the food component with reference to our abdominal cavity as well as the gastrovascular cavity. So renin is playing important role for what? In terms of the coagulation of the milk. Fine. So this is somewhat the condition when we are talking about the fluid Mosaic model. So you could often write this. So if you like the work with reference to the education as well as the lectures as well as the videos, so you could often subscribe, like as well as comment below in the text. Thus, I will be knowing that what concept actually you are hungry for or what haven't you understood or either got. So very very thank you. With reference to what? With reference to the RAHUL ANAND tutorials. So very thank you. Please like, subscribe.